This is the first of two lectures on genetics, or the patterns of inheritance. So for this first lecture, we will be talking about the law of segregation and the law of independent assortment. So for Mendel's law of segregation, you need to know the terms I've listed here. You need to be able to explain what the law of segregation means and determine the phenotypic and genotypic characteristics or probabilities for a given cross. So in previous lectures, we've talked about alleles, and we're going to revisit that concept for this lecture. So remember that the technical definition of an allele is an alternative or an alternate form of a gene. I'm going to write that definition for you here. In this image, the gene we are looking at is for flower color. There are two versions of that gene. That would be purple flowers and white flowers. So the gene is flower color. There are two alleles. We'll give big P to purple. Little p equals white. So all genes have at least two alleles, and some genes, like the ones for blood type, have more than two alleles. Now, when we talk about alleles, you need to understand what the terms dominant and recessive mean. And again, we talked about this in previous lectures. Now, I have this on the next slide, but I'll go over it here. So a dominant allele means that the trait or characteristic that is coded for by that allele will be present if that allele is in the genotype. So I wrote here the corresponding trait. In this case, it would be purple flowers, is exhibited if the allele is present in the genotype. Now, in contrast to that, the recessive allele means that the trait that it codes for, in this case, white flowers, is only exhibited if the individual is homozygous for that allele. So here we have shown capital P codes for purple flowers, lowercase p codes for white flowers. So you will only see white flowers if the individual organism has little p, little p. So let's go on to the next slide and talk about the terms homozygous and heterozygous. So I mentioned that all genes have at least two alleles. So here we have genotypes and phenotypes and the alleles that are included in each. So first of all, a genotype refers to the alleles that are present in an individual. So you can think of it as the letters that I'm using here. So genotype refers to the alleles present in an individual organism. Whereas the phenotype refers to the physical traits. So think of phenotype physical. Remember that you can pause this video to write some of this down. Now, in the picture, I have 3 flowers. The genotypes are big B, big B, big B, little b, and little b, little b. The phenotypes are purple, purple, white. Now, I also have written down homozygous dominant, heterozygous, homozygous recessive. So think about the root of these words. Homo means the same. So homozygous means the individual has 2 of the same alleles. Now, an individual can be homozygous dominant. They have 2 of the dominant allele, or homozygous recessive. They have 2 of the recessive allele. So it would be big B, or little b. Heterozygous, hetero means different. So heterozygous is always big B, little b, or whatever letter it is you're using. You do not say heterozygous dominant or heterozygous recessive, because you have 1 dominant allele, 1 recessive allele. So an individual is homozygous dominant, heterozygous, or homozygous recessive. So homozygous or heterozygous refers to genotype, whereas purple and white refer to phenotype or physical appearance. When we talk about genetics, we are talking about the work done by Gregor Mendel. Now, he came after Darwin. So when Darwin came up with his theory of natural selection, he did not understand about genetics. He didn't know what alleles were. He had an idea that traits were passed on from parent to offspring, but he didn't know how this occurred. It wasn't until Gregor Mendel started his experiments with the garden pea plant that we understood alleles and genetics. So as I stated, he conducted experiments on the garden pea plant. And I'm going to show you how these worked with Punnett squares in just a moment. First of all, when Mendel or anyone crosses 2 organisms, in this case plants, the first cross is what we call the parental or pea generation. The offspring of that cross, those are in the F1 or first generation. And then the offspring coming from the F1 generation are called the F2 generation. We're going to go to the whiteboard to take a look at the crosses that Mendel performed. Okay. We are going to look at the gene for flower color. The dominant allele, capital P, codes for purple flowers, and the recessive allele, lowercase p, codes for white flowers. In the original cross that Mendel performed, or his parental generation, the plants are what we call true breeding, meaning that they are homozygous. So we have I'm actually going to change my letters to, let's see, what should we change it to? Something easier to tell apart. F. So let me erase this. I'm going to rewrite this for you really quickly. Capital F equals a purple flower, little f equals a white flower. That'll be easier for you to tell apart than P. So Mendel started with capital F on one of the flowers. The X just means it's a cross, and lowercase for the other flower. So he was starting with a plant with purple flowers and a plant with white flowers. They were true breeding because they are homozygous. Now, the first thing that you need to do when you do a Punnett square is figure out what the gametes will be, the gametes that are produced by the individual organisms. Now, keep in mind that plants have egg and sperm as well. So if big F are the chromosomes in the original plant, you split those in half during meiosis, meaning that the gametes could be a big F or a big F because it's the same allele. The gametes for the other plant could be a little f or a little f. And it is the gametes, the potential gametes, that you put in your Punnett square. So now we're going to do a Punnett square. So we will put the capital F and the capital F across the top, and we'll put a lowercase f and a lowercase f across the bottom. What I want you to realize on a Punnett square is each letter that's on the side or the top of these squares represents a gamete, an egg or a sperm. They are haploid. They have half the genetic information. Now, inside the squares, you will be joining these gametes just as would happen during fertilization. And what you're getting here are probabilities. So if you're looking at my Punnett square, it has 4 squares. That is not to say that 4 plants were produced. Let's say 100 plants were produced. This gives you the probability that each plant will have a certain genotype. So to do the Punnett square, we join up the letters in each box. So for this first square, it will be a capital F and a lowercase f. I always write the capital letter first. Now for this one, we bring the little f over because that's the row that it is in, and we bring the capital F down. So again, we have big F, little f. For the third square, the little f comes over, capital F comes down, and again, we have the same genotype. And for this one, we will also have the same genotype. So we are going to look at the genotypic probabilities and ratio. So remember that the genotype refers to the alleles or the letters. So if you look at the 4 squares in my Punnett square, look at the different types of genotypes the offspring can possibly have. And it is only capital F, lowercase f. Now what you will do is count the number of squares out of 4 that have that genotype. And it is 4 squares out of 4, which equals 100% So all of the offspring for the parental cross will have big F, little f. And then let's look at the phenotypic ratio. Phenotypic means physical. Since capital F is purple, that's your dominant allele. If there is a capital F present in the genotype, the flower will be purple. So all of the flowers in the F1 generation will be purple because they are all big F, little f. So 4 out of 4 equals 100%. So what this is showing you is if you start with a purple flower plant and a white flower plant that are true breeding, big F, little f, all of your F1 generation will be big F, little f, purple. That is what Mendel did, but then Mendel took it a step further, and he bred plants from the F1 generation with each other. So we have a plant, I'm going to put a space between here so it's easier to see. Big F, little f times big F, little f. Okay, if you look back here, all of your F1s are big F, little f. So if you're breeding those F1s together, you're going to be big F, little f times big F, little f. So now let's look at our gametes. So from this plant, the gametes can have a big F or a little f. From this second plant, it can have gametes that are big F or little f. So now we are going to draw our Punnett square for this cross. Now, it doesn't matter which individual you put across the top and which you put across or down the side, just as long as you keep the gametes together. Okay, so now let's fill in the squares, and this would represent fertilization. So just to remind you, in this first square, we have big F coming down, we have big F coming over, which gives us big F, big F. Here in the next square, we have this one coming over because that's the row you are in, and this F coming down, which gives you big F, little f. I always write the capitals first. The third square, this little f is coming over, big F coming down. So you get big F, little f. And then finally, our fourth square, you're still in the row with the little f. And then this little f is coming down to give you the genotype, little f, little f. So now we are going to figure out our genotype, genotypic probability and ratio. You need to look in the squares and see which genotypes you have. So we have big F, big F, big F, little f, and little f, little f. Those are the genotypes that I see in the squares. So how many squares out of 4 have big F, big F? 1 out of 4 squares. 2 out of 4 squares have big F, little f. And 1 out of 4 squares has little f, little f. So what this is telling you is that for every 4 individual plants, you have 1 that will be big F, 2 that will be big F, little f, and 1 that will be little f, little f. Those are probabilities. Okay, so they're approximate. The other way of saying this is that 25% of your offspring will be big F, 50% will be big F, little f, and 25% are little f, little f. One final thing you need to be able to figure out here is ratios. So in order to get the genotypic ratio, you take the numerator, that's the top of the fraction, and use that for your ratio. So we have a 1 to 2 to 1 ratio of big F to big F, little f to little f, little f. And that is telling you what I just said. For every 4 individuals, 1 will have homozygous dominant, 2 will be heterozygous, 1 will be homozygous recessive. So now let's look at our phenotypic ratio. In order to determine this, you have to look at the phenotypes that are represented in the offspring. So our first square, that would be purple flower because it has capital F. Second square would also be purple flower because you have a capital F. Third square, purple flower. But your fourth square is white because it is homozygous recessive. So you have purple, remember phenotype, physical appearance, in 3 out of 4 squares, and you have white so you have 1 out of 4 squares is white. So 75 of your offspring, 75% would be purple, whereas 25% would be white. And your ratio, though, is a 3 to 1. Now, you also need to know how to simplify a ratio. So let's say I had a ratio that's 6 to 2. You take the smallest number, which is 2, and divide it into the other numbers to see if you can simplify. So 6 to 2 would simplify into 3 to 1. So 2 divided by 2 is 1, 6 divided by 2 is 3. All right, let's go back to the PowerPoint. So this image is showing you what I just did on the whiteboard. If your parental generation is true breeding purple, true breeding white, 100% of your F1 will be heterozygous purple. If I breed 2 heterozygous purple together, my F2 generation will be 75% purple, 25% white. And this is showing you the F1 cross, giving you the F2 genotypes and phenotypes. From his work, Mendel developed the law of segregation. There are 4 statements, and we will go over each one and simplify it somewhat. His first statement is that there are alternative versions of genes that account for variation in inherited characteristics. So first of all, a variation in an inherited characteristic would mean flower color is the gene. The variation is purple or white. So all number 1 is saying is that there are alleles for genes, and that these alleles account for differences like purple and white. Okay. So Now, the second statement, for each inherited characteristic, an organism has two alleles, one from each parent. So in this statement, characteristic means a gene. So for each gene, you have one allele from mom, one allele from dad. I'm going to write that. Let's see, I'll write it right here. So you can go ahead and pause this video to write that down. There are two other statements that go with the law of segregation. So just to remind you, Mendel came up with the fact that every gene has alleles, and these account for variation in a trait, like purple flowers versus white flowers, and that for each gene, you get one allele from mom, one allele from dad. Number three, if the two alleles of an inherited pair differ, one allele determines the organism's appearance and the other has no effect. What this means is that there is a dominant allele and a recessive allele. And if you have a heterozygous individual, the dominant allele determines the appearance. Number four refers to gametes. A sperm or egg carries only one allele for each inherited character because they're separated during meiosis. So what that is telling you is that an egg will carry an allele for a gene and a sperm will carry the other allele. And then they come together during fertilization. The alleles are joined together again so that an organism's traits can be determined. So okay, so just to summarize, law of segregation, every gene has alleles, correct? One allele is dominant, one is recessive. The dominant one determines the appearance in a heterozygous individual. I believe number two was you get one allele from mom, one allele from dad. And then finally, number four, an egg carries one allele, sperm carries the other. When they come together during fertilization, you get the diploid condition again. I'm going to stop here and I will make another video for the law of independent assortment. Okay, I'm going to attempt to do that.